This method can help answer key questions in the neuroscience field, such as the functions of different types of neurons. The main advantage of this technique is that it makes it possible to reliably identify the different types of neurons in vivo with inexpensive and accessible materials. I will be demonstrating the procedure together with Shinji Muramoto, a technician, and Lanlan, an assistant professor from a laboratory.
To begin this procedure, gently pull out the steel tube for pressure control from the barrel of the holder. Broaden the hole of the pin seat side in the barrel of the holder by drilling. Then, place a stainless steel pipe in the hole. Next, insert a ceramic split mating sleeve for 2.5 mm diameter ferrule into the hole. Make a hole in an L-shaped rabbet and attach the barrel of the electrode holder to the rabbet with epoxy adhesive. Subsequently, attach it to a manipulator with screws. Prepare a custom-built head post by making the columnar circuit board spacer into a cuboidal shape with a milling cutter and cut it into 2 head posts with a saw. Then, flatten the bottom of the head post with a milling cutter. Before the surgery, sterilize all the surgical instruments and a head post with an autoclave. Clean the stereotaxic frame and the heating pad.